an international journal of plant research and biotechnology, published by Springer. Now, we would like to start our today's program. Ma'am, Principal Ma'am, please grant the permission to start the program.
Yes, you are allowed to proceed, please.
Thank you, ma'am. With the permission of Principal Ma'am, today we start our program. Now, I request Professor Iksha Purak to welcome our invited speaker and all the participants present in this web series.
Thank you, Kanak, for inviting me and to the organizing committee of this International Web Lecture Series 2021 for providing me the opportunity to convene this International Web Lecture Series 2021 on Botany Made Easy, organized by Department of Botany, Ranchi Women's College, Ranchi, in collaboration with Society for Plant Research, Vegetos. We are privileged to have with us Dr. Samsun Nihar, Principal, Ranchi Women's College, Ranchi, as patron. She has rendered support and motivated all faculty members of Botany Department for organizing this 3-day web lecture series on Botany Made Easy. Myself, as you all know, Professor Iksha Purak, former head Department of Botany, Ranchi Women's College, and coordinator of biotechnology, and vice president of Society for Plant Research, acting as convener of this meet. Welcome you all. Dr. Snigdha Kumari, assistant professor and head, Department of Botany, as coordinator, and other faculty members of Botany Department as Sushma Dasguru, Dr. Salini Mehta, Mrs. Anita Tuddu, Dr. Pranita Pathak, and Dr. Chaya Thakur. All assistant professors are members of organizing committee, and I thank them all for their cooperation and support. We are fortunate enough to have a panel of eminent speakers as Professor S.K. Bhatnagar, Founder, Secretary General of Society for Plant Research and Chief Editor of International Journal Vegetos, Dr. Lakshmi Narayan Mishra, Mrs. Simpla Mehto, and Dr. Anita Maan with us in this web lecture series. This International Web Lecture Series 2021 on Botany Made Easy is a 3-day program from 4th to 6th October 2021. Today is the second day, and in this second day, the third lecture will be delivered by Mrs. Simpla Mehto, Research Associate, Indiana University at Bloomington, Indiana, USA, on electron microscopy protocol. She will discuss everything in detail. I am glad and proud to share that Simpla Mehto is our alumni, second batch biotech honors student, I think 1997 to 2000. She
Yes.
was very hardworking and had keen interest and zeal in learning. After joining Indiana University, she has gained expertise in the field of electron microscopy. Welcome, Simpla. This lecture series will certainly provide innovative ideas to make the subject interesting and easy, and students will be benefited by interaction. I would like to welcome all esteemed speakers and express my gratitude on behalf of Ranchi Women's College and Society for Plant Research. I am glad to find that large number of faculty members of different subjects, research scholars, and students have joined this 3-day International Web Lecture Series 2021 in Google Meet and also in YouTube live programming from Ranchi Women's College channel. I welcome you all and wish and hope that our students will certainly be benefited by attempts of organizing such programs from time to time. Thank you all for patient listening. बोलो.
Thank you, ma'am. Now I request Pranita Pathak, our Assistant Professor of Botany Department, to kindly introduce to the speaker of today's Mrs. Simpla Mehto.
Respected Principal Madam, patron for this event, Professor Iksha Purak Madam, former Head, Department of Botany, RWC, and Vice President of SPR, Professor S.K. Bhatnagar Sir, Chief Editor, Vegetos, Society of Plant Research, Dr. Anita Mehta Madam, Dr. Snigdha Kumari, Coordinator and HOD, Department of Botany, Dr. Susma Dasguru Madam, Assistant Professor RWC, our Honorable Speaker, Mrs. Simpla Mehto, all the dignitaries, participants, my colleagues, and my dear student. I am Pranita Pathak, Assistant Professor, Department of Botany, RWC. A very good morning and warm welcome to the second day of the International Web Lecture Series 2021 on Botany Made Easy, organized by Department of Botany, Ranchi Women's College, in collaboration with Society of Plant Research, Vegetos. It is my pleasure to introduce today's speaker, Mrs. Simpla Mehto, as she is the alumni of our college, the Ranchi Women's College. She did her undergraduation in 2001 from Department of Biotechnology, RWC. I would like to mention one more thing. At present, it is 10 a.m. It is a morning hour, but in Indiana, where our honorable speaker lives, it is 12:30 a.m., which is a midnight hour, and she is awakened only to give her lecture for the benefit of our student. I thank you, ma'am, for accepting the invitation and for this encourageous gesture. Simpla Mehto is currently employed at Indiana University in the lab of Dr. Andy Gelhoff. She has experience in the field of microscopy for more than 15 years and is adapted in using transmission and scanning electron microscope and confocal microscope. She has done her MSc from MS Gaur University Sagar, MP, India in 2003. In 2009, she has got the degree of MSc in Molecular and Cell Biology from University of Alabama, USA. She is a great scholar as she got the scholarship for higher education during 1994-96 from Hindustan Copper Limited, India. In 2003, she is member of Taylor Committee Society of Protostologists. In 2007, she is a student's representative at the Department of Biological Science in Alabama, USA. During 2008-2009, she got Graduate Council Fellowship from University of Alabama, Tuscaloosa, USA. Madam has a teaching experience from 2005-2010 at University of Alabama, Tuscaloosa, USA. She has great skill in techniques like molecular biology, general lab techniques, etc. Apart from that, she is expert of management and leadership skill and computer skill also. Madam has published 10 research papers in journals of high impact factor like plant genetics, developmental biology, and nature communication. She has done many valuable poster presentation also. She has done a lot of training in Ranchi and abroad also. She has handled projects like to study adsorption of vaccine for hemorrhagic septicemia in ALOH3 gel at Institute of Animal Health and Husbandry, Ranchi, Jharkhand, and project like Master Use of Amira for the construction of 3D image of the oral apparatus of tetrahymena thermophily at the University of Alabama, Tuscaloosa, USA. Once again, madam, we are very grateful for accepting our invitation and we look forward to your talk on electron microscopy protocol. Over to Mrs. Simpla Mehta. Welcome, ma'am.
Thank you. So I can start now. So switching tabs, I cannot see any more other people. So hopefully everybody can see my.
Yeah. No,
Okay. So
presentation is visible and
so
audible also.
okay,
So you
that's
can
good then.
start.
Yeah. So today I'm going to give a talk about electron microscopy, like protocols, discuss a little bit, and let's see. I am targeting mainly students because professors must be having some idea about all those things. Since everything about me was told, so there is nothing much to discuss about in the slide other than the fact that I just wanted to say that I started my career doing biotechnology. So mostly I was involved in bacteria, study of bacteria. But then when I moved to Alabama, I started studying tetrahymena, which is a protist. And after that, when I moved to Indiana University, I started working with Drosophila. So in my career graph, I have moved from microbiology to eukaryotes and been have done other organisms also, but this is what my stretch of model organisms that I've worked with. And being said that, I wanted to imply that to all the students that please do not feel that you are in one organism or you're a botanist or something. I would like to say we all are scientists and we can all move from one model organism to another because there are many ways to answer questions of science. So with that, I want to just give a hint of what we are going to talk about. Of course, I want to deal a little with the history of microscope because I think so there are a lot of scientists who have put a lot of effort into developing the microscope field of microscopy, and I think so spending 2 minutes of our time acknowledging them is a respect we give. And I want to talk about light microscope, of course the development principles and everything. In general, I'm hoping that the students that who are listening to this talk have a basic idea of how light microscope works and everything, because what my understanding of the field of microscopy is that if you understand one microscope, you can switch to other microscopes because the basic principles are same. It's just that we have adapted to different techniques based on the research questions we have. And after that, today I'm just going to deal with transmission electron microscope because talking about too many microscopes, I think so I'll overload the students with too much of information, and I think so that will lead to losing attention of the kids. And if needed, we can of course have some other day to talk about other kind of microscopes also. Saying that, seeing is believing. This is what I believe, and that's how I got interested in the field of microscopy. For me, to be able to see things was a proof that things exist. And it's just a very cute story I would like to say about myself. When I was in high school, 11th, 12th, I thought, I could see DNA. It took years and years of education, higher education. And it was actually when I was in University of Alabama that I actually started reading research papers. I realized that we cannot see a DNA. It's always all the proof that DNA, the double helix DNA exists, is indirect proof. But by that time, I had developed the interest of being able to see things. And that's why microscopy interests me a lot, because that's where you can visualize whatever you're imagining, you can actually see, and you have a proof of it. So, being said that, what is a microscope? It's an instrument with which we can view objects that are too small to be viewed by naked eyes. We can see houses, we can see trees. But when we have to see bacteria and viruses, which are very small to our eyes, we use microscope. So microscope has a very long history, and it has been, the field has grown with lots and lots of effort of great scientists. But these scientists were basically not biologists, they were more physicists, because they understand the concept of lenses and they understand the concept of light. And so the first effective microscope was developed by Janssen and Janssen, a father and son. And I'm showing you a picture of the microscope that they developed, which, they use 2 or 3 variation combination of lenses, and that's why it's called as a compound microscope. And of course, after that, Anton von Leeuwenhoek was there, who developed another kind of a microscope, which was single lens microscope. And then again, it was Hooke who combined all the efforts of Janssen and Leeuwenhoek and developed a compound microscope and added stage, which is what the precursor is for today's light microscope that we see. But as the turn of century, on the 20th century, actually, it was Zeiss and Abbe who have greatly contributed to the field of light microscopy. They pushed the limits of light microscopy, and we could get the maximum amount of magnification that was possible using light as the visible source. So, when we talk about all the different kinds of microscopes, there are 3 terms that are very important that we need to understand, because these are the 3 things that defines any kind of microscope, whether we talk about light microscope, we talk about electron microscope, or anything. So the first one is magnification. What we see through a human eye that's a standard. That's why we called it 1x. So anything which we see through the lenses will be multiplied, and we see 5x times, 10x times, and 20x times. So that is the concept of magnification, which is we are enhancing the size, like the image of a sample or a specimen at certain times, and so we are able to see it. And then another concept is resolution. Resolution is conceptually, what resolution means is the ability to distinguish 2 objects as different. For example, let's say that we look at a mountain. When we look at from, like maybe 2 kilometers away, we see it's one mountain. But as we come closer and closer and closer, we see that perhaps there are 2 or 3 different peaks. So the ability to see that one mountain can be 2 or 3 different peaks is called as resolution. And of course, when we are talking about biology, the contrasts, the ability to see, because the background is white. Let's talk about a bacteria or a virus or even a cells. They're all transparent. So they, like bacteria, it isn't water. Water is transparent, and the bacteria is there, which is transparent. So we cannot see a bacteria in the water. So what we have to do is to be able to distinguish or give some kind of a contrast to the bacteria, to its background that is water, which gives the ability to see. So these are the 3 things that scientists have been fighting, struggling, adjusting, and developing all the kind of microscopes that we see today. So again, just a little bit of introduction of light microscope. I want to stress a lot on light microscope in the beginning, because what I've realized that either it's a light microscope or electron microscope, the basic format or the basic plan in which a microscope is built does not change. The principles which is based on which a light microscope is based is the same as what electron microscope is based on. So understanding light microscope will give or help student to understand electron microscopes better. So in any kind of a tabletop compound microscope that most students in college and universities have access to, we see that there are two parts. One is illumination, that is the light source, and the imaging system. So let's look at this compound microscope, the tabletop microscope here. This is called the illumination system, because this is the light source that which we use. In general, nowadays we are using bulbs with white light, and earlier time we used to use sun as the light source. So what happens, the light comes out, is emitted from here. This is the stage where we put the slide with the sample. So what happens, the light goes from here, and depending on which kind of an objective lens that we have, the light passes through there, and here there are a bunch of other kind of lenses through which, like, it passes through like this, and the image, the specimen which is here is enhanced or magnified or become bigger. And once they become bigger, like let's say this is the size that is achieved based on 10x magnification, then it's going to be, the image is going to be produced as 10 times more than the specimen. If it's a 40x, then it's going to be 40 times. And of course, see, so this one does 40, and this ocular lens most of the time has about 10x of magnification. So what we are seeing, that we see image of the specimen which was here, and we see a magnification of about 400. Let's say this one is 40, the objective lens, and this one is eyepiece. So 40 times 10 is 400. So a bacteria that is there, or the cell which is there on the slide, is 400. The image that we see is 400 times bigger than the actual size of the cells. So can you imagine what kind of a help the microscope is? Because now we are able to see which we could not ever visualize with naked eyes. So when we go for any kind of a light microscope, which I think so all the students in botany must have done, in undergrad I did some onion cells and all those things. We have a basic protocol that we follow. That is, in any kind of a sample prep that we do for light microscope, there is a fixation of cells, and then we process the cells in whichever way. Every protocol is different. And then we of course do any kind of embedding, which is like putting in the parafilm. And after that, we have to, once it's there, then we have to make thin slices of it, which is called as sectioning. And after that, we have to stain it, like put, all kinds of dye and this and that to develop a contrast. And then we look under the microscope. My stress about saying these steps in light microscope is that you would be amazed and surprised that the same thing is what we do in electron microscopy also. So the rules which are there for light microscope stands tight even for electron microscope. So once we understand light microscope, I think so we are at least 10% or 15% into understanding electron microscope also. So now what happened? Like we see light microscope. Of course, as I said, we can do up to a 60 or 100 to like 1000. I think so. With light microscope, what we can achieve is about 1000 times magnification. But was that enough? Well, it was not enough for the biologist, but that's where we were at the turn of the 20th century. And let me tell you why it's very important to understand that why we um, have, like, we cannot, the limitation of having any kind of a um, like magnification where it was solidified. Okay, this is where the light microscope ends. For that, like, the limiting factor was light. The visible light that we use as a source of illumination for light microscope has a wavelength of about 400 nanometers. Remember and Zeiss, who made and perfected the compound microscope. Abb's principle was that you can get a resolution of half what is the wavelength of the illumination source. So, saying that, when we know that the visible light is about 400 nanometer, that means what we can get is the resolution is about 200 nanometer. So 200 nanometers, so anything which is closer than 200 nanometer in a cell or anything will look as one point in a light microscope. So that's why when we are trying to balance the amount of magnification that we can do, like adding lenses on top of lenses to increase the magnification, what happens, it becomes very important that we have a limitation of magnification, because after 200 nanometers, 2 points cannot be distinguished as separate points in a light microscope. So when this happened, people started looking at other options. Of course, the scientists knew that light can be used as a source of illumination, and it has a wavelength of about 400 nanometers. Then what can we do to increase the resolution? So Zeiss started using ultraviolet rays as a source of illumination. So what it did was, it since the wavelength of ultraviolet is smaller than that of the visible light, it improved in the resolution of microscope. And doing that, the Noll and Ruska, who were physicists of Germany in the World War time, they thought, why not use electrons? And they used electrons as the source of illumination, and that's how electron microscopy was developed. So now, what do the advantage of electron microscopy? Of course, we can get a good resolution, and since we can get good resolution, we can add more and more and more number of lenses, and that will help in getting the magnification higher also. So just to say here, the wavelength of electrons is 2.5 picometer, that is 10 to the power minus 15. So imagine where we are talking about in nanometers, and now we are at picometers. So theoretically, with electron microscope, because the wavelength of electrons are so less, the resolution is like the two things, anything which is inside the cells, even two molecules, can be distinguished as two separate identities, and that how much powerful an electron microscope theoretically can be. But of course, things are never easy. So, but there are issues with lenses and all those things which has reduce the resolution of electron microscope. But as science is developing in the field of microscopy, scientists are working harder and harder to increase the magnification and have a balance between magnification and resolution. So when we talk about electron microscopes, there is basic two types of electron microscopes that's available. One is called as transition, and one is called a scanning electron microscope. The top one that you are seeing here is one of the newest version of the transmission electron microscope that is available in the market. It's from the company JOL, and I think so I'm very fortunate enough that I am able to work on this microscope. The other one here that we see is a scanning electron microscope. Now, let's not go into too much details of how they are different, because they are definitely different. But the basic one liner difference between electron transmission and scanning is that with transmission, the electrons are able to pass through the specimen, and that's how they are able to provide image for people to view. But when we are talking about scanning electron microscope, it does not penetrate the specimen. The electrons move on the surface of the specimen, and whatever reflection and diffraction that we get on the movement of electrons from the scanning, when the electrons hit the surface of the sample, is used for imaging of the specimen, and that's what we see. And at this point, earlier, like till I guess 80s or even till late 90s, film based images were produced. But now with computers, things have become easier, and we have digital imaging. So that's why we have computers here on which we can see the images that is there on the stage. And yeah, so it's faster, easier, and more convenient. Microscopy is a lot more easier to do compared to maybe 30 or 40 years ago when it was film based. Now, seeing all these things, today, as I said, I don't want to talk too much about too many things, because then everybody starts losing their attention. I want to focus today's talk on transmission electron microscope. And I think so this is going to help, because moving from light to electron to scanning, what we will see that they have all basic principles same. There are basic idea of any kind of microscopy is same. So it should be beneficial even just to focus on transmission electron microscope that I am going to do today for the talk. So again, it was 1931, Nolan Ruska developed the electron microscope. You can see in this image the original electron microscope that we have. So this is the first that was developed, and today here we are, so much more smaller and lot more efficient and better with all those things. So in this electron microscope, of course, they used electrons as the source of illumination, because remember, that's what the scientists were thinking that time, that the source of illumination, which is from light, we have to move to UV or to something else, which will help for better resolution. But is it that everybody just jumped into electrons immediately? No. All the scientists during that time were looking at a lot of other options as the source of illumination. They tried neutrons, they tried X-ray photons, and they tried electrons. Electrons turned out to be the most efficient because it has a specific charge in it, which is negatively charged. Since electrons are negatively charged, what advantage that has is that something which is negatively charged can be channelized, or it can be targeted, or it can be pushed in a certain path through an positive potential. With neutrons and X-ray photons, that was a little difficult to do, because neutrons are neutral and photons are neutral. So that's why electrons turned out to be the winner of using as a source of illumination. And then what other advantages does electron have? Electrons are negatively charged, And what they do is, as anything which is negative will have the tendency to stick to something positive. So it is a very strong interaction with the matter. And of course, electrons are they are much more smaller than the photons and the neutrons. So what it does, that it can probe into a small volume and it generates a very strong signal. I will explain how the signals are developed by electrons in a later slide. And as I already mentioned, electrons are charged particles. That's why it's very easy to use um, different kinds of like positive forces and magnetic forces to put them in a certain path, and which is like um, so the beam, or the illumination beam can be better controlled. So, saying that, we now know why we have electron microscopes now and not neutron microscopes. I mean, we have some small neutron microscopes and X-ray crystallography, but basically for general uses, it was electron microscope. So here I want to have a basic schematic diagram I have for the electron microscope. I'm basically talking about transmission. Let's be very specific for the whole talk from now onwards. I'm not going to talk about scanning. So when I say electron microscope, please consider it to be a transmission electron microscope. So whenever we look at any electron microscope, what we are seeing that on the top here, it's the filament, which is going to be, nowadays it's mostly lab 6, which is the source which produces the electrons. So when the electricity or the power cable hits here, this is where is the electrons produced. Remember, in a light microscope, what we are seeing, that the power or the light source is in the bottom, but in an electron microscope it's in the top. That's how they have designed it, because it will go down. So that's some kind of a basic difference, but let's not focus on the differences, but try to understand that either it's a light microscope or an electron microscope, we have an illumination source, which is here the electrons. After that, when electrons are released from the source, they will travel in every direction. These are waves. We cannot control in which direction it will go. It will go everywhere. But the whole idea is that the waves are going in every direction, we cannot get a proper image. For that, we have to streamline the illumination source, and we have to streamline the electrons. And that's what these magnetic lenses do. These are magnets, huge, very powerful magnets. What they do is make sure that the electrons which is coming out from the filament is pushed in a straight line down here. Now, what we see is, we on the path of the electron, we have this area which is where we put the sample inside. And when the sample is here, which the electrons hit, and after that, what will happen? They will travel here and there. But there will be some electrons which pass through the specimen. And based on what electrons have passed through the uh, specimen, they're again focused properly with another kind of magnetic lenses. And this is where the screen is, which is mostly fluorescent screen, where the image is formed. So remember, in a light microscope, we had light in the bottom and the eyepiece on the top, and we were seeing image. Here, the light source is on the top, or the electron source is on the top, and we see the image in the bottom. But what we are trying to get the concept here is that electrons or light pass through the samples and we get an image, and what we are actually visualizing is not the specimen but the image of the specimen which is magnified. And that magnification, based on the type of microscope we are using, can be from 1,000 to 10,000 to 100,000. So this is the principle on which the TM is based. So remember, 1931 was when Knoll and Ruska were able to make the electron microscope. But it was 1945 when Keith Porter, a great scientist and cell biologist, was able to produce the first micrograph, which I have depicted here of a eukaryotic cell. So what took us 15 years? Whenever physicists and scientists, when they make instruments, it's basically more for material science people, because what they do, they take any kind of material, they put it in the microscope, and voila, they get some nice images and whatever studies they need to do. But when it comes to electron with biology, things are very different. Of course, Keith showed us that the transmission electron microscope can be used to study cells and tissue structure and functions which are at really nanoscales and very small. But the path that we had to go from using the microscope was a long path. It has lots of challenges, and there are some things that I would really like to stress about. When we look at an electron microscope, let's see here. This is the small part that we are seeing, but around it, we have a very huge column and made of very strong metal. What it does, it creates vacuum. There is, because the whole concept of electron microscopy is that it works under vacuum. So now, when I said that Knoll and Ruska discovered electron microscope, and material science people were very happy because they could stick in their samples and get images. But for biologists, that's not easy. That's why it took us 15 years, 15 long years to able to get the first picture from an electron microscope. Because the biggest thing is, any kind of living cells have water in them. So if there is water, we cannot take that sample into a very high vacuum area and try to get images. What will happen? All the water will be sucked out of the cell. So the cell will lose its structure. And of course, the water will contaminate the electron microscope, and it will lose the vacuum, and all kind of technical difficulties we start having. And then, of course, another problem was that life is made up of basic elements like carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. These are all very small elements. When we say it's a very small element, let's think about chemistry. What do we have? They have a very small atom. There are very less numbers of electrons. And so then what does that mean? That means that any electron that is going through the cells will just pass through, because there is nothing to stop them. Here I want to stress on the fact that how is that important, because or maybe later I can talk about it later, how electrons work here. And so when we are talking about electrons traveling through the whole chamber of electron microscope, like in a transmission electron microscope, as I said, electrons are scattered everywhere. And of course, electrons have a very small wavelength. So based on that, we can only observe very small, or let's say, to be very precise, a very thin object can be observed under a transmission electron microscope. So it's like sometimes, We can say like a whole bacteria is also sometimes very difficult or too big to observe under a transmission electron microscope. So looking at all these difficulties, sample prep was um, a challenge, and it took 15 years of lot of scientists with lot of effort and lot of knowledge to able to prepare samples for transmission electron microscope. So when we are trying to do a sample prep for transmission electron microscope, the we have developed lots of protocols. So there are two things which is very important, that there is no point in observing a specimen which is not in its native state. When I say in a native state, that means if a cell, the way it is in the body, when we take it out, like make a section and look under the microscope, it should remain as same as the natural it is in the body. Otherwise, we will get false data. So therefore, when we do all kind of fixation protocol, we have to stress on the fact that the cells stay in its most native state. And the second thing that is very important, remember I said that we need our samples to be dry. There should be no content of water, because if there is a content of water in the sample, it will give false results, because water inside a vacuum will evaporate, it will shrink the sample, and of course the microscope will not work properly. So now we understood like what are the technical difficulties with the sample we have and what is our goal. So based on that, again, there is a protocol for sample preparation which is same as that of any light microscope procedure. That is, we have fixation of samples, and then we have a processing of sample. In processing, we have to do dehydration, and after that infiltration, and we have to do embedding, which all comes under processing. And then of course we have to do sectioning, and after that we visualize it under the transmission electron microscope. So now I will try to go into detail step by step of how a sample is prepped in a transmission electron microscope. I am giving a very general idea. This is nothing to do with very specific organism. So when we get a concept of how chemicals interact with each other, how they are used for a sample prep, and why they are used for a sample prep in an electron microscope protocol, um, mostly these are very standardized, and we can go back and forth from bacteria to Drosophila to plant cells. But of course, every lab has little bit of different tweaking, and every person does little bit of things differently. And of course, when we are talking about perhaps a plant cell or an animal cell, sometimes some things work differently based on the cell structure. But today, what I'm going to talk about generalized, which is what any protocol will follow. So the first step is fixation. Why do we fix? The samples must be stabilized to preserve the structure of the living tissue with no alteration from the living state. So that means we fix the cells because we want it to be in the exact native state as it is in nature. So how do we achieve this? There were scientists who tried lots of different kind of fixatives, but the most effective one is called as Karnofsky fixation protocol, which uses a combination of glutaraldehyde and paraformaldehyde. Every lab, every sample has to be treated little differently, but most of the time a combination of glutaraldehyde and paraformaldehyde works as a fix. So generally for me, what I do is like I make a mix of glutaraldehyde and formaldehyde, put my samples into it, and leave it overnight. The idea behind using two fix is we have formaldehyde, which is like a very small molecule. So remember, any kind of a living cell has cell membrane and different kinds of membranes and cell wall and all those things. Formaldehyde will immediately enter because it's a small molecule. It enters through the membranes very fast and it starts forming all kinds of cross links. But these, especially with the proteins, and these are little reversible. So what it does, that it, the purpose is using formaldehyde, that it immediately goes inside the cells and starts forming cross link. Then glutaraldehyde is the one which is strong, fixative, and it forms good chemical bonds with proteins and like and cytoplasm, so inside the cytoplasm. But since I have put here the picture of the chemical structure of glutaraldehyde, it is a bigger molecule. So what it does, it takes longer time to enter the cell membrane and into the cell. So generally, we, depending on the thickness of the sample, the primary fixation needs to be done at least 2 hours, and it is most effective to go overnight. We don't want to fix too long because overfixed cells are also not good. So we have to have a balance between less fixing, more fixing, to being an appropriate fixing. And that comes with experience on and how everybody likes their samples to be different, like how they treat the samples. After we do any kind of fixation, which is like overnight, it's a very basic understanding in any kind of protocols that we do, that we have to wash the samples. And when we do washing of the samples, we use buffered solution. The whole idea of using a buffer solution is that any kind of cell, the cytoplasm has a certain pH and they have a certain constituents. And to maintain it, we just don't want to put any kind of a solution which will lead to leakage of the molecules in outside the membrane or that kind of thing. So when we put a buffered solution, it maintains the pH of the cell so that things are not leaking in and out. And also what it does, like in um, EM protocol, there are lots of, when glutaraldehyde and formaldehyde, they form bonding, there are lots of other kinds of radicals which are released, and the buffer balances those kinds of radicals. So once we have done the primary fixation with glutaraldehyde and formaldehyde, and after that we do washing, it's very important to remove all the chemicals which was there. So that's why we go for multiple washes using a buffered solution. Mostly that we use is cacodylate buffer. And after that, we use osmium tetraoxide as the second fixative for EM samples. This is where it perhaps can be a little different than light microscope, where only perhaps I'm not very sure how other people do, but generally one fixation is good enough. But with EM, we need to do um, a secondary fixation, because what, remember, glutaraldehyde and formaldehyde, they are trying to bind, they do cross-linking with proteins. But any kind of cell has lots and lots of lipids and all those things which need to be stabilized too. So for that, we use osmium tetroxide because it binds with the lipids and it stabilizes the cell membrane of the cells. With osmium, just let me say that any sample, when it reacts with osmium, they turn black. So the first step of primary fixation, everything will look white or green based on whatever color your sample is. But after that, osmium tetroxide will react to those things and make everything look black. When we're talking about protocols, it's very important to understand the chemicals and to understand the safety of using those chemicals. Glutaraldehyde, formaldehydes, osmium tetroxides are fixatives. That means they are going to fix cells. So we want them to fix? Not necessarily. They can fix anything. So that means they can fix us also. We are living, our hands and legs have living cells in them. So if we are exposed to glutaldehyde and osmium tetroxide, we will get fixed also. Therefore, following all kind of safety procedure is very important when we are dealing with these kind of chemicals. Wearing gloves, and osmium especially is very, very, very toxic. There is, like in US, everybody cannot use osmium tetroxide to do samples. We have to be specially trained, and mostly in labs there are people who are licensed to be able to deal with osmium tetroxide. And all the processing that we do is mostly we do under the hood, because osmium tetroxide releases lots of gas, which if we breathe in, of course, then it's going to start fixing the cells in the nose. And eventually, people have said that if you're not careful, you lose the capacity to smell. So, just to let you all know, it's very important to follow safety procedures when we are doing this kind of sample prep. So, after we do a fixation of this with osmium, again we go for washing, and we do washing with buffer. So the first goal of sample prep is done, that we have been able to fix cells in its native state best of our capabilities. Now the second goal is to remove all the water which is inside the cell, because remember, with the cell water we cannot use it under the microscope. So this starts the process of dehydration. D means removing, hydration means water. So what is the best way to remove water? We start putting the samples through a graded series of organic solvent like ethanol and acetone. Each have their advantages and disadvantages. Some prefer acetone, some prefer ethanol. Me, on a personal basis, I have always used ethanol. So what we do is we put the samples like in 10% ethanol, then 30%, then 50%, and 70%, then 100%. And all this process, we do it at room temperature. And the timing that we use for dehydration, of course, it is dependent on the size of the sample and what protocol you're following and how it works for you. And so once the dehydration happens, some in some lab they use the final dehydrant as polypropylene oxide. Some people do, some people don't, but it helps in ensuring that any kind of ethanol which is there, which is old, and it must have absorbed any kind of water, and it's no more 100% ethanol, then it has not effectively removed all this water from the cells. Then at least when we add propylene oxide, we ensure that the 100% water has been removed from the cell. And again, propylene oxide is flammable, very toxic, and you should not be inhaling the fumes of propylene oxide. Another important reason why we want dehydration to be a very good job, because the next step that we do is infiltration. Remember, like in light microscope, we are trying to put the samples in paraffin. Here, what we do is we put them in resin. And this is again very important step, because we want the water to be removed, but we have to have a balance that we do not do too much of dehydration, because then that will lead to shrinking of the cell. So balancing all that, once we finish dehydration, we go and we do infiltration. For infiltration, what we do, instead of using, like in light microscopy, we use paraffin. In electron microscopy, we use resin. Resin in a layman language is plastic. So what we do is we take different kind of chemicals that are mixed together and we make a liquid plastic. These in mostly we have to make uh, what you say is like every lab makes fresh plastic, it's liquid. I've given you example here that mostly people use. One is epoxy category, in which there is a epon plastic which has a kind of a its own combination of ingredients. Then epon araldite is there, and then there's another one called as spurs. So these use different kind of chemicals that are mixed together to make liquid plastic. And that is, epoxy is generally good for doing regular TEM, but sometimes what happens is that we need another kind of research questions in which we are doing immunogold labeling or something, which is more like immunochemistry kind of a thing that we do. And in those cases, we use acrylic resins, which is like LR white, LR gold, or Lovacryl. Again, these are all liquid plastics that we have to make in the lab. It's just different combinations of different kind of chemicals that we do. So epoxy, one big different reason I said, why we use epoxy and acrylic. And another thing why epoxy is used for general TEM sample prep, because it is a thermosetting um, chemical or resin. What does that mean? Thermosetting means, thermo means heat and setting means it sets in heat. So generally when we have to solidify the plastic, we put it in a temperature and it solidifies. So it's very easy and convenient to work. So again, when we are talking about infiltration, once since I've said about all the ingredients that is there, the way infiltration works is that, remember, we are doing dehydration, so the samples are in 100% ethanol or they are in propylene oxide. Now what we do is start infiltration. Resins or the plastics are very thick liquid. So what we do is start doing graded infiltration. So from 100% propylene oxide, we'll add 30% resin and 70% propylene oxide. Then we go 50% resin and we do 15% propylene oxide. And then we do 100% resin and we do 2-3 changes. The idea here is that of infiltration is that the resin will go inside the cell and it will let all the propylene oxide outside. So that, remember what the idea was, any kind of cell has water. The water is, in the dehydration process, the water is moved out of the cell and it is replaced by ethanol or and propylene oxide. And now what resin will do, it will go inside the cell and it will remove all kinds of ethanol and that kind of thing. So once that is done, infiltration is done. Now we do embedding. Embedding is a process in which, here we see these are called as molds. So these are what you say. Here what we do is we pour the plastic here, which is of course liquid. Most of the time it is epoxy liquid. And then what we do is we put the sample on the tip here. If you can see on these are the black things that you can see. That's the sample. Once we put the sample here, and after that we put it in a oven where it at least at 65 degrees for 48 hours for it to solidify. For remember, that's why epoxies are called as thermosetting polymers or resin, because when we give heat, they all become solid. And they're different. This is one kind of a mold that we use. There are other different kinds of molds that can be used based on the research question and the need of the lab. Here, just to mention that I am stressing more on epoxy, because acrylic resins, they are not thermosetting resins. That means heat will not let them solidify. When we use Lovicryl, LR Gold, LR White, they are acrylic resin. So the way to solidify those resin or those plastic is by UV light. So we put UV light on those, we put the like plastic in these molds with the samples, and we put this mold under UV light and leave it for 48 hours or something, and then they become solid, and they're very hard. And at this stage, these samples can stay with you for eternity. So that's how it is. Again, it's electron microscopy, so everything has to be toxic. All the ingredients that we use for making resin are toxic, and they need to be worked under the hood. But luckily, once we have these plastics, once it solidifies and it has gone through the process of thermosetting, they become non-toxic, and we can use them with bare hands, and we will not have any kind of issues. So now, just going back, I just want you all to see the samples that is here, because after this we are going in processing of the sample that we have. The thing that we do is trimming the blocks that have been generated after embedding. It has to be trimmed in a certain way, and I'm not sure in histology how people do it. I personally, I did not trim when I had to do some samples using paraffin, but in electron microscopy, we have to trim for transmission electron microscopy, especially because your sample needs to be really, your specimen sample needs to be really, really, really small. When we're talking about the sample size, it is about 1 millimeter or something. So these blocks that you see here, they are big, and these are about um, 2 to 3 millimeters. So extra plastic, which is around the sample, is moved. So when we're talking about this image here, let me go back and show you. We are talking at the tip here, or tip. This tip is looking upwards, or it is looking at your face. When it does that, what you do is, let's say the sample is here. We start trimming all the extra plastic which is around the sample. So the way they start trimming is cut this area, and ultimate goal is that we want to have a trapezoid shape, flat surface where the sample is. Square shapes are a little difficult to work. So trapezoid shape is the most standard, which that we trim the blocks into, because that helps with the sectioning. This can be achieved in two ways. Some people can do it manually using a razor blade, and some people can use a glass knife to do all the trimming. And if you have lots of money in your lab, you can use a diamond knife. Because let's talk, how I'm talking about the difference on the cost is, a razor blade would cost us perhaps, like $5 for one razor blade, and a glass knife costs around $3,000. So, sorry, I mean, a diamond knife cost us $3,000. So that's what we are looking at. So most of the time, people do maximum amount of trimming manually using a razor blade, and in the end, we all end up using a glass or a diamond knife to do the last finishing touches of the trimming. Once we do trimming, that's where things starts getting very complicated. Till now, we have processed the samples, and it's all fixing using chemicals and all those things. Lot of knowledge about chemicals, how they behave, how they interact is important, because that helps in a good sample prep. But it is not necessarily you have to be expert. You can do if you're given a protocol, and most of the time people end up doing right. unfortunate, sometimes people do some mistakes in the fixing and all those things. You have little bit of leeway. But now comes ultramicrotomy. When we talk about ultramicrotomy, this is where, as a microscopist, your expertise lies. The better you can section, the better images you will get. Fixation takes care of, like 25 to 30% of, credit for getting a good image. And ultramicrotomy takes another, let's say, 50%. So this is where it has a huge learning curve. People have to spend years to get expert into it, but once you get it, the results are beautiful. As I had showed, in light microscope, you use, and paraffin embedded things are, like you use microstat to do the sectioning. The same concept is here in ultramicrotomy. This is a Leica ultramicrotome. Leica is a pioneer in providing microtomes. These are very nice nowadays and very convenient to use. Again, when we're looking at a microtome here, nowadays everything is like computer based. So this is here where you get to control a lot of things and a lot of automation happens. And this is where the stage is, and this is the ocular lens through which you look at the sample when it is doing. If anybody can see, this area here has been expanded to this. So let's focus on this part. This is called as the chuck. And chuck is where it holds the sample here. It's a round kind of a knob thing inside which the sample is held. And this is a diamond knife. The diamond knife, everything is not made of diamond. This area where the knife, what is called as the cutting edge, is made of diamond. And this whole thing here is called as the boat or trough. So what we do is we put the sample here, and then we start, sectioning, and we get thin sections. Here, to just get some idea on the what is ultramicrotomy. When we're talking about ultramicrotomy, the sections are in nanometers. The thin sections are somewhere around 20 to 30 nanometers, and thick sections are around 80 to 90 nanometers. They are real thin samples. So as humans, we cannot visually see these these sections if it's in the air. So what we do is we fill this trough with water, and then when the section floats on it, like you can see here, the sections are floating, and that's how we visualize the sections through this lens. And here it's a very abstract science, but it's how do we, of course everything is automated. So when we set the ultramicrotome and doing sections at 60 or 70 nanometers, it's going to do 60 or 70 nanometers. But it's not, sometimes things happen. Like when we are doing ultrasomy, it is very much affected by the environment, the room temperature, if it's cold or not. And this instrument is very sensitive to vibrations. So if there is little bit of vibrations, your sectioning will be affected. So, when we are doing sectioning, it's like we don't breathe very heavy. We don't move too much. Our body is very still, and we are breathing very slowly so that sectioning is not affected. But in general, rule of thumb is that how do you decide if the sections are good or the thickness is good? Since we see it in the water, the sections have colors. If it's very thin, it's going to be white color. And if it's little thicker, it's going to be silver color. And then it's gold, bronze, green, blue, purple. So general rule of thumb is that if you have light gold color sections, they are good for the transmission electron microscope. So when you are sectioning, you should be able to section light gold color sections. So now as I said here, we have all the sections floating in the water. Now, how do we pick up the sections? This is another skill set that is acquired with steady hands and practice. Here I'm going to show you one method because this is what the picture I was able to get. And I personally do not follow this protocol. I do something else to pick sections. But in general, we have this metal loop that is available. So what we do is we take this metal loop and press it on the sections. So now what it does, this metal loop will carry a water, and in the water on the surface, the sections will be there here like this. Now we take this section, this loop, and we put it on the grid. When we put it on the grid, what will happen? The grid will be here, which is like a slide that you have in a light microscope. This is a slide for transmission electron microscope, and we called it grid. So we put the grid and then we put the loop with the water and section. But it is such amazing that nothing happens to the water most of the time if you put it like that. So then what we do is we gently take a paper, and then we start taking the water out. Like you see, it's a Wattman filter paper. What it does, we start sucking the water slowly out of it. And once that is done, the sections is transferred to this grid and we can take the loop out. And this is how you go back and forth all the time to pick sections from the diamond knife and put it on the grid, which is the slide of transmission electron microscope. So here we have one kind of a grid. So based on so I'm just showing here example that, like this is what a light microscope slide looks like. And this is what a transmission electron microscope slide looks like, which is called as a grid. And grids don't necessarily have to be this shape. They can be this shape. The grids can be smaller, bigger. They can be this shape. They can be this shape. All the choice of grid is dependent on the fact what is the research question and what and how we want to do it. These can be like 200 grids, then there can be 200 holes here, there can be like some 150 holes, and these grids come in material like gold, nickel, and copper. So based on what kind of questions we have, we use the kind of grid we want. And the material, like, do we want to use a gold grid or we want to use copper grid? But for general purposes, copper grid is the best because it's cheap, it's a lot more sturdier, and it does the job. Here this is a grid box. So it's just to get the size comparison of how big the grid is. This is where we put the grids inside this. This is a grid storage box. So imagine this is the slide and this is the grid which goes here. So that's what we are talking about in size when we talk about a light microscope and an electron microscope. So everything is really, really ultra small. So like these grids, we cannot hold with hands. We always need forceps and all those things to move it around, and those are really fine tip forceps that we need. So now we have processed our samples, we have sectioned our samples, and now what we have to do is stain. Remember, any kind of biological samples that we have, by nature, they have low contrast. Of course, when we are doing fixation, we have added osmium tetroxide, which gives a little bit of contrast because it starts making things black. And so it starts having some heavy cross-linking with lipids and all those things. But that is still not enough when we look at under the microscope. So we need more processing, that is basically adding contrast, and that is we do through staining in any kind of microscope. When we do light microscope, we have, like gram negative stain, positive stain, methylene blue stain, and then toluidine blue staining and that kind of thing. In EM, there is a very standard protocol that we follow for most of the time. That is urinal acetate and lead citrate staining. Again, not to mention urinal acetate and lead citrate, they are very, very, very toxic chemicals. So, urinal acetate is a radio isotope. So, again, we have to deal with them with a lot of caution, lot of training is needed, and any kind of disposal or wastage that is generated has to be disposed in a very strict protocol. So, in general, urinal acetate stain is made either in water or it can be made in ethanol. Some people make 2% urinal acetate, some people make 4% urinal acetate. There is no standardized people have not gone more than 4%. So let's say we stick between 2 to 4% of urinal acetate in either ethanol or it can be in water. So again, we take all the care of making the solution, and it has to be made one day before. It is preferable to make fresh stain solutions, but with experience now we can make solutions and it can go for a month or two if we take care of it. Urinal acetates are light sensitive, so we have to keep them away from light even when we are storing the stain and even while we are doing the staining process. If we do it in the light, the staining does not do very well. So urinal ions bind to proteins and lipids with sialic acid carboxyl groups, and so it stains with the DNA and RNA, and so it is a positive stain that we are doing. So once we have done with urinal acetate staining, we do another staining which is called as lead citrate staining, which is the combination of lead citrate that is given slide is like we mix lead nitrate and sodium citrate, and we use CO2 free water to make the solution, and then we optimize the pH. And this protocol was developed by a scientist named Reynolds, and that's why it is called as Reynolds lead citrate stain. So we use the stain so first step is staining the grids with all the sections with urinal acetate, and then we stain it with lead citrate. And lead citrate staining has to be done in a CO2 free environment. If there is carbon dioxide present, the lead citrate reacts with the carbon dioxide and we get some kind of precipitation, which we do not want on the sections, because then it creates artifacts which we don't want. So here I'm just trying to give example of how the staining process is done. So we add in a petri dish, we add a paraffin and we put drops of urinal acetate. Then with the forceps, we take the sample, the grids out, and we put it on the drop. Generally, we leave for about a minute or two on the urinal acetate, and during that time, it is covered completely, and and we put a box or something so that it is dark. So once that happens, after 2 minutes, we remove this grid, for like holding this grid with a forceps, and we take the filter paper and remove all the extra urinal acetate from the grid. Then we add drops of water, and then we put the grids on the drops. And that's how the washing of the grids is done. After that, again, what we do is we take lead citrate and we put them in drops, And see, we have added sodium hydroxide pellets on the side. What sodium hydroxide does, it sucks up all the carbon dioxide which is around so that lead citrate does not precipitate. Again, we'll leave the um, grits and for lead citrate for some time, and then we can wash. So what I explained earlier is one method of washing in which we put drops of water and let the grits pass through this. And some people try to have beakers of water and you just pass the grits through it, dipping in and out, like. But this is a not very effective process because we have chances of losing the sections when we are dipping in and out of the beaker. So here again, the staining is done. And then after we finish staining, we put the grids back in the grid box. Uh, it is preferred that after staining, we wait overnight so that the stains dry, so there is no water content on the sections, because remember, we don't want water to be in the transmission electron microscope. But we have to make sure that once we stain it with lead citrate, it is not exposed to CO2, because even after all the sectioning, all the staining has been done, there are chances that um, the grid can react to CO2 which is present in the environment and cause precipitate. So usually if people have to wait, what they do is they finish the urinal acetate staining and then stop, and then do lead citrate staining after some time, leave it overnight for the sections to dry, and then go to the microscope for observation. So here, this is the most exciting part of the TM, is that you are in the microscope facility looking at the huge microscope and trying to use the grid. And so this is one big process that we have to do, is learn how to put the grid in the sample holder, and then how to put the sample holder into the microscope, making sure that we do it properly, because it's a very, very, very delicate instrument. And if little bit of mistake and the vacuum seal breaks, and we cannot use the microscope, and it takes few hours for the vacuum to again build up and use the microscope. So this is called as the sample holder. This is the only area which is outside the microscope. So this is where we can hold it. It is called the holder. We never touch this part because it has to be as sterile as possible, because this is the part which goes under the microscope. And especially from here to here, we are never allowed to touch for any reason, because this is where all the beam and all exposure things will happen. Now, if anybody can see, there is a small hole here. This is where we put the grid. So giving a closer look, this is where the hole is, and this is where the grid will go. So what we generally do, every company has a different way of making sample holders. This is one sample holder in which the clip is taken out. There's a switch here which removes the clip. We put the grid on top here, and then we put the clip back and lock it. This is basically Hitachi does that most of the time, as far as I can remember. And this is an innovation which recently happened, which JOL has done. I'm not sure if other companies have done. This is called as a quadrat holder, in which what it does, it's like a flap. These can like open, and we can put 4 grids at one time and then close it. And so it's easier in the sense that we don't have to put one sample in and take it out and then put second sample in, and then we can put 4 samples in together and we can view them. Once we put the samples in the grid and lock it, we have to ensure that this is locked, because once vacuum kicks in, if things are not locked, they open, they break, grids get lost, which is not good for either us as a scientist, because we don't want to lose the sample. It is not good for the microscope because we don't want debris inside the microscope. So this is here where we put the sample holder in. After that, the basic protocol generally works is once you stick it in, then we twist this one, and then the vacuum slowly starts pulling this part inside into the microscope, and it locks. So again, let's come back to a schematic diagram of transmission electron microscope and see where we are. So here, remember, we have the electron gun, which emits electrons. And of course, we have here the lenses, which is targeting all the electron beam directly here. Now the sample holder is in here, and it comes directly in the path of the electrons. Once that happens, what it does, the electrons will pass through the samples. And when they pass through the samples, they get deflected, diffracted, reflected. But whatever is passing through it is again channelized by these magnetic lenses and is brought on the here, which is the like the screen where we can see it. And this is the lens through which we can see the samples, and sometimes we can see the sample directly here. And this image is also projected to the computer, and we can see it in the computer here. So now when we talk about this, we have to understand how image formation takes place in TEM. What we do is when we put the samples inside, like this, the stage here where the electron beam is hitting the sample, remember we have used uranyl acetate, lead citrate as the staining agent, which binds to different things in the cell to form bonds. Now we remember living cells are made of carbon, hydrogen, oxygen, nitrogen, sulfur. These are small elements, and most of the time the electrons passes through them, so we don't get contrast. But when we're talking about heavy metals like lead citrate and uranyl acetate, what is their kappa? See, their atomic number is 238 and all those things. So what it does, it has tons and tons and tons of electrons in them, especially in the inner shells of the atom. So what happens is that when the electrons which come from the beam and hit the samples, they are interacting with the electrons of the uranyl acetate and the lead citrate. Like they are trying to bump each other, and they are trying to push here and there, because negative and negative will definitely, they will repel each other. So when they're pushing around, lots of different kinds of energy is produced. Those are, and because of that, electrons lose their energy and they gain energy, and all those things happen. It's a whole lot of physics happening here, and based on those kind of energies that are released, which is tapped, and again through all the magnetic lenses are sent to the screen, and that's how we see an image of a sample. So this is how we see an image, and we get beautiful images, but it's not always that happens. Now, remember, in any kind of microscopy, especially with electron microscopy, because it's such a big machine with a lot of vacuum and all those things, main goal is to preserve a cell in its native state. And what the image we want is in the native state. If we get anything less, it's of no use. So if we get an image that is not necessarily a good data, we should be able to distinguish between good data and bad data. And there can be lots and lots and lots of criteria for bad data. Like we talk about fixation. When we look at this image, okay, this looks that, okay, the membranes have been preserved properly. But if we get something like this, then we know the membranes are not preserved properly. That means that's not a good image. That is not an image based on which we can interpret data, we can design our experiments, or we can publish. Similarly, when we are not able to maintain the osmolarity of the cells, the buffers don't work effectively, or the fixation is too much or too less, we have shrinking of mitochondria, Golgi apparatus, and all those things. And those images are again not good. Here, again, I'm showing here one image here where the infiltration did not work properly. The resin which was supposed to go in properly did not go, and we have all the holes here and all that kind of thing. So when we put this under the microscope, because of the high energy of the beam, the film is going to spread or shrink. They will behave in different ways, and the image whatever we get is not a good data. So I'm sorry about this image. It's really not clear. I was not able to do it. And so, but I want to explain at least what are the artifacts which is available, which are present here. So earlier in the slide, I talked about all the fixation artifacts that we can have. And then let's talk about ultramicrotomy artifacts. So when we are using for sectioning, it's preferred to use diamond knife because they are really, really sharp. But it's very expensive. As I said, we spend around it's in Indian money, we can talk about anything between 2 to 3 lakh rupees for one diamond knife. And life of a diamond knife can be from anywhere from 6 months to 3 years, depending on the person's skill set of use and how much it is used. So some people use glass knife for doing sectioning. Then what generally happens, because the resin is a very, very hard material, what it does, it breaks the edge of the knife. So if that happens, then you start getting these lines that you can see here. When these lines are there, which is called as knife marks, it can even happen with diamond knives, but it takes a little bit longer than glass knives. So these knife marks are there. If it's one or two, those micrographs are acceptable. But to have a really good publication quality micrograph, we don't want knife marks to be there. And then here you can see, if it's a really bad knife, how you are getting lines and lines of knife marks, which you don't want. Then, of course, as I talked about, that when we do microtomy, we do sections around 30 to 80 nanometer. But sometimes, if you are not careful, or due to whatever reasons your sections are thick, let's say instead of being light gold color, it is blue or purple color. And that's when what happens, like the electrons which is hitting the samples or the specimen on the grid, they are not able to pass through in a very straight path. So then what happens is that you cannot get a very crisp focus image. All the membranes look very blurry, and the reason is your section is too thick. And then there are another kind of artifact we have during ultramicrotomy, which is called as chatter. So when chatter happens, when for some reason there is a vibration, the vibration can be on the chuck, the arm that is moving, or like if you're sometimes the air in the room. So if people open the door, like open or close door, that kind of thing. So those chatter is like waves. You see on the section, it looks light dark, light dark, light dark. And those kind of micrographs are unacceptable as a data. And of course, when we say there is water contamination also, when you get strings like this, remember we are picking up sections from the water. We are collecting sections on the water. When we are using water at different stages of staining, the water needs to be really clean, filtered water. We use 0.22 micron filters to get clean water, and when we do any kind of these kind of processing, because when this happens, we cannot know whether we of course know it's water, but it's not a proper data anymore, and it cannot be used for publication. And next stage is staining. Staining is again where we can get a lot of artifacts. I said, like we don't necessarily need to stain images because osmium has already is there when we do the fixation, but this is how it looks. So we don't get very clear um, pictures, but perhaps it can work, but generally it is not a good micrograph. So when we stain it, the same thing looks so different. So this is the kind of a good micrograph that we are going to use for any kind of a publication. So here, when you see all these black dots, these are called as lead citrate deposit. Remember, we for lead citrate to work effectively, we need a CO2 free environment. So if you're not able to provide that little bit of CO2 here and there, and it precipitates, and these are dots that which is there, and that's not good, because when cytoplasm, when we're talking about different kinds of molecules, or molecular interaction between some two molecules, or we are looking at RNA or something like that, even the small dot can give a false data. So we don't want any kind of lead citrate deposits on a micrograph. And of course, urinal acetate, we have to make sure that it is in solution, it has not precipitated out, and it's like not exposed to light, because when all these things happen, this is the kind of dumping of urinal acetate happens, and this data is when you get a picture like that, it is completely useless. So it's very important that we are very cautious from the first step to the last step, because everything is very important to get the end product, and things can go wrong all the time. So with that, this is a slide I put just for all of you to see where the future of electron microscope is. There are a lot of variations. There is like STEM, which is a scanning transmission electron microscope. So basically what it does, it's a transmission electron microscope where we are using thin samples and everything, but when it uses the imaging process, it follows the principles of scanning electron microscope. And of course, I had talked about, X-rays being used as a microscope. Those are like the EDX. And then we have energy loss spectroscopy. There are many different, like there are some tomography and all those kinds of microscopes available in the range of electron microscope. I have really not used any of these to have much deeper idea about it. It was just a slide I wanted to put up for people to just get an idea that there are many things available other than a transmission electron microscope, and people use these microscope for very specific research questions, like. And of course, when we are talking about TM, it has not yet perfected. Scientists do hope that we can do better, and have better and better transmission electron microscope. Remember, I said that when we talk about the wavelength of electrons, they are about, what I was saying is about 2.5 picometers. So based on that, we know that the resolution of a TM is infinity. We will be able to see any kind of protein to protein molecular interaction, or anything that is happening inside the cell through a transmission electron microscope. But in reality, are we able to do it? We are not able to do it. remember when we're talking about resolution and when we were talking about magnification, all these things are juggle and balance that we have to follow. So to get, maximize the resolution of a TM, people are definitely working on how to make lenses and how to have better electron sources and how we are able to accelerate the speed of electron to a certain where it does not destroy the sample, but it's able to go and do all kinds of things. So this is where the future of TM research is. And after that, this is the end of my talk. I don't know how long I have talked. I lost track of time. I didn't see. I hope everybody is still awake and I have not crossed the time. So with that, I would like to thank Dr. Andy Zellhoff. He has, for 10 years, I've been working with him. He is one of the best mentor who has given me the freedom to do research. And fortunately, he is very well funded. So I get a lot of opportunity to use a lot of good microscope. I don't have limitations of, facilities or the supplies that need for electron microscopy because as I said, it's a very expensive science. It's not a cheap um, a technique or the that we can have. And of course, my lab members who have been supportive. I really, really appreciate Icha ma'am. I am lucky. I was her student. Being taught by Icha ma'am has been a joy, a privilege. And thank you so much, Icha ma'am, for pushing me, for giving these kind of talks, because as usual, I try to run away from these things. And Anita ma'am, she is my aunt, relative, and she has made sure that I do come back to my college, meet with my professors and teachers who have taught me and who made me what I am today. Had it not been Anita ma'am, it's really difficult. So thank you. She's my aunt. So it's a privilege. She was my teacher too. Tissue culture was what she taught me. And I would like to thank Karachi Women's College. I mean, at this point, 20 years down the lane, I really don't know that many faculty members. Things have changed, or whether all principal and everything have changed. Sushma ma'am is still there, who was new when I was about to graduate from Women's College. And there are other ma'am, I'm not sure. So, but it's a privilege. It's a pride for a student when they can come back to their own college where they have learned to be able to talk, interact with their professors as peers, and be able to give knowledge to the next generation. It's a payback way of doing and I get this opportunity, which I don't think so many of my friends are able to do it. And I appreciate that from Women's College and to all. Thank you so much. I hope everybody's awake.
Thank you, Simpla, for such nice comments and such a very, 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 very good presentation. And I hope my students will be benefited a lot. Thank you, Simpla, again.
Thank you, ma'am.
For being with us and for accepting our invitation. Now over to Kanak.
Excuse me for a second. Simpla, congratulations for such a wonderful and good lecture. And this is not only pride for you, it's pride for us also, for the pride of the college that our student is presenting over here. So I wish all the best for you for future journey also. Thank you.
Thank you, ma'am.
But Nagar sir, do you want to say something? But Nagar sir को mic खोलना पड़ेगा उनको. कहना है उनको.
सर आपका माइक म्यूट है.
मैं कर दे रही हूँ उनका.
I simply wish to congratulate Simpla for a wonderful lecture, and I was online every time, and I enjoyed and learned a lot about electron microscopy, TEM, and all that. And I'm sure that she'll be defending her PhD and will be serving her own institution sometimes in future. And as a founder of Society for Plant Research, we had been practicing these type of lectures, and it is naturally a knowledgeable event where the students and the teachers as well, like me, a person who has retired 3-4 years back, and Professor Icha Purak, we are long associates. Since 1989, we are associated together as a member of Society for Plant Research and as founder member. So I congratulate you, Simpla, for a very, very wonderful lecture, and God bless you. Thank you.
Thank you, ma'am. Now I request to share the questions in the chat box if they have any. Ma'am, we have one question from Iksha ma'am.
Mhm.
Ma'am, please discuss about ma'am, please discuss about volume microscopy and its application and its protocol.
Yes. So, I mean, this is something, yeah, we have talked about that. I wanted to, this is something I'm doing. It's a very recent microscope, which is like 5-10 years ago that has been, and it's getting optimized. It's a scanning electron microscope. So, basic idea about it is, like it works as a scanning microscope, but what it does, it provides us with a data which is like a transmission electron microscope. So what it does, it has a microtome which is inside the microscope. So what it does, electrons go to the sample, they hit, then it does a raster kind of a movement, and we get an image. And then again, a microtome will go and cut the top slice. Then again, it will take the image. Then again, the microtome is going to cut, and then it will take the image. So what it does, that we get serial sections, which doing in ultra microtome is really difficult. So that's a very automated process that happens in the microscope. And what we get is images like we can have 50, 100,000, depending on what we have. And then once we get those images, we use Amira as a software to align them and develop a 3D model of something which is like what we see in a transmission electron as a 2D of an image. Like the image that we get is two-dimensional, that we see X and Y axis. Let's to be very clear. And what we can do with the volume scope is align all these images. And when we start from the top and we go up to the bottom, we can get a 3D image, in the sense we can understand how long a neuron can be, or how long the cell is, what is the size, what is the shape, in a very, very exact way. And the protocol, again, it's simple. It's similar to what I have already discussed, but it has lot more detailed tweaking needed. And I thought today was not a good time, because if I start talking about 3 or 4 microscope, people will get bored. So I would request Icha ma'am to give me some another opportunity, maybe like 2-3 months down the lane. I can talk about scanning electron microscope, and that's when I can discuss more about how to work with the volume scope, and I can show my data there. Because in Indiana University, other than the facility manager, I am the one who does volume scope in whole of Indiana. I'm one of the specialists in doing volume scope. So I'm still developing the technique, still learning software and everything. It's a process. So yeah, that's an introduction of volume scope, and details can be in future, I hope so.
Thank you, Simpla, for giving the answer, and we will again invite you. You must be ready that you will be
Yes.
invited again and again on our platform.
Yeah. This time I was complaining that I did not get enough time to prepare the lecture, so I'll start preparing from tomorrow, so that next time I can be ready for any time.
And Dr. Icha, I wish to point out one thing, that this time is a torture for those who are staying in USA. It's really a torture because
But that is
in the
her fortune
because
also. And that is
in
her
the morning she has
privilege
to
also.
attend to her duties. She has to attend to her lab, and it is almost 2 o'clock, I suppose.
It's
And
2:30, but next time I will be smart. I'll make you all wake up
Punishment
at
तो
7
होना
o'clock
चाहिए ना
in the
इसका
morning.
कि मैडम
This
का
time I
स्टूडेंट
was
होने का.
considerate. So
you,
next time my
you,
talk
She
is
is
7
my
o'clock in the
student
morning.
and she must get some punishments like this.
no, why punishment? Because she has entertained you, she has given so much of knowledge, so punishment is not desirable. So,
But I love
thank.
Icha ma'am. I love Icha ma'am. I can do anything for her.
Yeah,
I
yeah,
mean,
yeah,
so
yeah. She is a lovely lady, I know, and a very passionate teacher. Mm. Thank you very much
yeah.
once again. Thank you.
Thank you. चलो शुरू करो आगे.
Now, I request Raita ma'am to brief the lecture and present the vote of thanks.
Heartiest thanks, madam, for giving such a wonderful, informative lecture. The briefing of this lecture includes Madam has explained about the working principle of electron microscope and its similarity to the working of light microscope developed by Jensen and Leeuwenhoek. She has explained about the history of microscope in a simple and in a very interesting way. In this lecture, she discussed about the development of electron technology to discover new frontier of the living world. She explained about the transmission electron microscope, its protocol, its advantage, and almost everything about transmission electron microscope needed to know. She also described various methodology to successfully navigate the process of developing a publication quality micrograph. Once again, I congratulate you, madam, for this successful deliberation. And on the behalf of the Department of Botany, Ranchi Women's College, and Society of Plant Research Vegetos, I would like to congratulate you, madam, for this successful deliberation and PowerPoint presentation also. It would be very useful for our students. I would like to thank our principal madam, Iksha madam, our eminent speakers, and all the members of organizing committee for organizing this type of informative international web lecture series. I'm sure our student will be benefited to to be part of this. I would like to thank all the dignitaries and participants for their valuable presentation. Thank you. Over to Kanak.
Thank you, ma'am. I may request all the participants present here to kindly gather for the last day of international web lectures. Tomorrow we have Dr. Anita Man, Division of Crop Improvement from ISR, Karnal, Haryana, and the topic for lecture is advancement of molecular markers for plant research. I may also request all the participants to fill the feedback form which is shared in the chat box and different WhatsApp groups. Lastly, I would like to take the permission of Principal Ma'am to end the program.
कहाँ बोली प्रिंसिपल मैडम को. ठीक है तो एंड कर है ना.
End करें?
Principal madam, the program is over. ठीक है. We can leave the meeting.